Hello again. I'll talk now about macrolides, which is the antibacterials for the fourth, final, and shorter lecture in the series of antibiotic lectures. These are the learning outcomes, and I'll talk about them as we go through the presentation. Initially, these antibiotics isolated from actinomyces, and the semi-synthetic macrolides have excellent pharmacokinetic properties and stability, and that's why they have been given a greater attention. Picromycin was the first macrolide antibiotic was reported, followed by erythromycin and carbamycin. In Iraq, the most common prescribed macrolide antibiotic is erythromycin. Here is the erythromycin base of the erythromycin antibiotic, which is made from a large lactone ring in black with either 14, 15, or 16 carbon atoms, depending on the type of the macrolide. For example, erythromycin and clarithromycin are 14 carbon structure, while azithromycin is 15 carbon structure, as we will see later. The structure also contains a ketone group and linked to amino sugar group in blue color, as you can see in this slide. Examples of macrolide antibiotics are erythromycin, clarithromycin, and azithromycin, which are the most common used in clinical practice. They are orally active, and some of them are bacteriostatic, which prevent the growth of bacteria, and others are bactericidal, which kill the bacteria. These antibiotics are commonly prescribed for respiratory infections and act by inhibiting the protein synthesis when they bind to the 50S ribosomal subunit, as I'll explain later in the mechanism of action. The first example and the most important macrolide antibiotic is the erythromycin, which consists of 14 carbon units in the lactone ring, as shown in the structure ahead. The view on the right hand side shows the binding of the erythromycin in red to the ribosomal subunit RNA in dark green. In regard to the mechanism of action of macrolides, they work by inhibiting the bacterial protein synthesis, as I mentioned before, when they bind to the 50S subunit shown at the top of the figure on the left. Normally, and as you know, the protein is synthesized from amino acids and polypeptides, and the peptidyl transferase enzyme adds the peptidyl attached to the tRNA or transfer RNA, and macrolides inhibit the peptidyl transferase as well as a translocation, and as a result the polypeptide chain will be disrupted and the bacterial protein synthesis will be stopped and terminated. Now we'll talk about the issues associated with macrolides in general and erythromycin in particular. The main two problems of the macrolide antibiotics and erythromycin is the poor water solubility and acid instability. To solve the first problem and to improve the solubility of the erythromycin, the salt form of this antibiotic is produced and the dimethyl amino group is introduced into the sugar group, and the whole amino sugar system in blue color at the top of the structure attached to the lactone ring enhances the solubility of the drug. I'll talk now about the second issue, which is instability of the macrolides in acidic stomach. The reason behind instability is the formation of hemiketal and ketals as a result from the reaction between ketone and alcohol, as we will see in the next two coming slides. Before I talk and explain the formation of hemiketal and ketals, I would like to draw your attention to the structure of the erythromycin base, which contains ketone group in red at the top left and two hydroxyl groups in red and blue, and ketone and hydroxyl groups actually play the key role in the formation of hemiketal and ketals that cause acid instability. So the formation of the hemiketal, which is formed by the reaction between the ketone group in red and the alcohol group in red color as well, in the presence of stomach acid. The structure on the left showing the erythromycin base and ketone and hydroxyl group before the reaction with acid, and the structure on the right showing the link or the connection between the ketone group and the alcohol or hydroxyl group in red to form the hemiketal. The type of this reaction is nucleophilic by the addition of the OH nucleophile in red to the ketone group. However, this reaction is reversible and the drug is still active, as the second alcohol group in the blue is still not involved in this reaction. So the formation of hemiketal, which is formed by the reaction between the ketone group in red and alcohol group in red color as well, in the presence of stomach acid. The structure on the left showing the erythromycin base and ketone and hydroxyl group before the reaction with acid, and the structure on the right showing the link or the connection between the ketone group and alcohol or hydroxyl group in red to form the hemiketal. The type of this reaction is nucleophilic by the addition of the OH nucleophile in red to the ketone group. However, this reaction is reversible and the drug is still active, as the second alcohol group or hydroxyl group in blue is not involved in this reaction. Now we'll talk about the formation of ketals, and if the hemiketal produced in the previous slide, which is shown now on the left here, reacts with the second alcohol group in blue, then the ketal will be formed on the right of this slide, and this is irreversible reaction and the drug becomes totally inactive, so the gastro-resistant drug or the coated form of the drug must be given or administered. However, to avoid this problem and to prevent the formation of ketal, the reaction between A and B or between the hemiketal and the second OH in blue must be avoided by blocking either A or B side. For example, introducing ether group in a place of alcohol group in red to form clarithromycin, as you can see in the next slide. I've selected clarithromycin and azithromycin as two examples of macrolide antibiotics, and I'll talk about them. In relation to the clarithromycin, it consists of 14 carbon atoms in the lactone ring, as you can see in the structure ahead. To prevent formation of ketal in clarithromycin, alcohol or hydroxyl group OH in red is blocked as an ether or 6-methyl ether shown inside the red circle, so the clarithromycin is acid stable. Another advantage of introducing 6-methyl ether is producing methyl group, which decreases the polarity of the drug and enhances the pharmacokinetic properties in terms of better absorption and larger half-life. In terms of pharmaceutical uses, clarithromycin is used in soft tissue and respiratory infections with activity against gram-positive bacteria. Comparing to clarithromycin, which consists of 14 carbon atoms, azithromycin consists of 15 carbon atoms, and the ketone group was replaced with amino group in red to avoid ketalization and to improve acid stability and pharmacokinetic properties such as half-life. So, azithromycin is usually taken orally once daily due to the long half-life it has. Similarly to clarithromycin, azithromycin used for respiratory infections and has a broad spectrum against both gram-positive and gram-negative bacteria. Finally, and in relation to the toxicology of azithromycin, it disrupts or increases the heartbeat and causes tachycardia when taken with antihistamines such as astimizole or terfenadine. Also, a patient takes azithromycin may experience nausea and vomiting due to the poor toleration. So, at the end of this lecture, you are expected to know the macrolides in terms of classification, structure, mechanism of action, and also the issues related to these antibiotics regarding the acid instability and toxicology. And as usual, this is the main reference book, and thank you for your attention.